Hello students and welcome to my third and final screencast for this biochemistry unit. This part will be on enzymes. Just like all the others, it is not for profit, it's meant to help you. I like to think of enzymes like this ladder right here, okay? It helps my son get up so he can brush his teeth. Without it, he couldn't do it. All right, and so here, I love these PJs. It has little enzymes on them, little science stuff on them. All right, I'm going to use a lot of Khan Academy content. This is slightly different than the Google slideshow I posted in classroom because that one was from Miss Foglia, who was a renowned AP biology teacher. She unfortunately has passed on, and she's left a great legacy and is a really awesome slideshow. We've done some of her labs throughout the year. Okay, from the study guide, what should you know? So why don't you pause the video and see what you got? Okay. So let's go over the first one. Describe how enzymes speed up chemical reactions by lowering activation energy. So this is a enzyme kinematics graph showing how a reaction happens both with an enzyme and without an enzyme. So look at the graph. Can you do the I squared? What do I see and what do I interpret from it? And so hopefully you see that an enzyme is going to lower the activation energy for a reaction to happen. This reaction might happen anyway, right? But it's going to make it happen a lot faster. A great example of that would be the lab we did in class with catalase. Now, we haven't done it as of this recording, but we will. And hydrogen peroxide. So catalase is an enzyme that breaks down hydrogen peroxide, which is H2O2, into water and oxygen. That would happen normally. That's why hydrogen peroxide is in brown bottles, so it doesn't get broken down by sunlight. But the enzyme makes it happen a lot faster. If you want another example that's like a sports-based one, right? If you're playing football, your cleats will help you run faster. It takes less energy to get good grip by having cleats. Imagine trying to play in flip-flops versus cleats. This is from Khan Academy, same image is showing the reaction progress, the energy. The ability to interpret graphs is really important. Try and use that I squared strategy. Right? So here I see that with an enzyme, often called a catalyst, right? Enzymes are biological catalysts. It helps to speed up a reaction. Okay, describe the lock and key theory of enzymes. Well, I told you it's kind of like going to your house, right? The keys to my house are only on my keychain and my wife's keychain, so they open up my house. And so enzymes are specific, right? This substrate is only going to fit with this enzyme. So you have thousands of different enzymes in your cells. Now, some enzymes are more specific than others, just like some keys could jiggle into different locks. But let's not lose sight of the big picture, right? Enzymes are specific for the reaction. It's like a lock and a key. The new version of that is called the induced fit model, which we'll go over in class. So what's happening in this picture? Let's understand. Here's the substrate. That's what's being worked upon. It lines up with the active site. That's the part of the protein that can hold the substrate in place. This enzyme changes shape slightly as the substrate binds, and then voila, it helped to break it in half or to form the product. Right? And it doesn't always have to be breaking things down. It could be synthesizing things together. So it's specific for each one. This is a really good picture for understanding enzymes. All right, here's just some of those key terms from Khan Academy. So the enzyme is a biological catalyst, almost always a protein. The substrate is whatever molecule is being worked on, and the active site is where it binds. So in our experiment, the substrate was hydrogen peroxide. That's what the enzyme catalase is working on. Couple of tips for you. Enzymes are specific. We just went over that. They're often named for the reaction they help. Lactase, we've learned about that, helps to break down lactose. Proteases help to break down proteins. Lipases help to break down lipids. Sucrases help to break down sucrose. And DNA polymerase helps to build DNA. So that's an example of one building up. So they're often named for that, but they're not always named for exactly what they do. And they often end in ASE. Remember back from the last video, OSE often signifies carbohydrates and ASE often signifies an enzyme. Okay, so that was what does an enzyme do? It helps to speed up a chemical reaction. They're very important in your body. Without it, we couldn't live. So now what happens if you change the temperature or pH on an enzyme? So think about your lab, and hopefully you should have some vivid understanding from that. But if you change it, say you heat it up, you can change the shape of the enzyme. It's called denaturing. Or if you make it too cold, or if you change the pH. What's happening there is it's really changing the interaction between all those R groups and those amino acids. If you remember the amino acids, they have specific R groups that help them fold into the shapes that they want to be in. And here, if we heat it up or cool it down or change the pH, it doesn't work as well. Well, do you have different pHs at different spots in your body? The answer is yes. Right? Your stomach's more acidic, your intestines are more basic, so they have different enzymes. Right? Pepsin works well in your stomach and in pH of 3, whereas trypsin works well in your intestines at pH like 8.5. Okay, and you should be able to remember that from the lab, right? Where we heated up the liver and then we put different stuff on. So you should be able to discuss that lab that we did in class. And in this, I'm not sure if we're going to get to it, honestly. So if it's close to the test and I told you don't worry about it, then you can cut the video off here. Except for you might want to better your understanding. Or maybe we will get to it. Right? But so enzymes can be inhibited. They can be stopped. Like if you had a bunch of defenders between you and the goal, right? They could slow down your progress for scoring a goal in football. So after you eat, you need lots of enzymes to break down food. Less so later on, right? When you're just resting or you're in biology class and you're not eating because you shouldn't be eating, right? You won't need as many enzymes. So your body helps to regulate it. You need more at some point than others, which makes sense. So you can regulate them with activator and inhibitor molecules. These are molecules that say go on or stop. Sometimes they only work with the presence of cofactors, so they need something there to tell them to turn on. A good example of that would be what's called the lac operon that we'll deal with later on. That's not in humans though, but it's a good example of an enzyme being turned on when lactose is present. And then some enzymes are inhibited by the end product of the pathway. So example would be cellular respiration. So making energy or ATP. When ATP is available, then it turns off the pathway. So many enzymes work in a step-by-step function. All right, here's an example. Okay, I'm back. So here's an example of a normal reaction happening, and here's a competitive inhibitor. This is from Khan Academy. It's blocking it. All right, here's an example of a non-competitive inhibitor. So it's going to bind to it and it'll either make the enzyme not work or it could change its shape so that the substrate doesn't fit with it. This is how a lot of poisons work, things like penicillin or cyanide, etc., and how pathways in your body work when they, a lot of them are in metabolism, when they don't need the end product anymore. So here would be an example of it. So let's say that here's enzyme 1, triangle comes, boom, it makes purple rectangle. Purple rectangle goes to enzyme 2, it makes this blue thing goes to enzyme 3, it makes the final product. That goes to the start of the pathway and turns it off. Now it can no longer accept triangle, it wants a different shape. So it's good, it's a way that your body can regulate itself, and it's called negative feedback. All right, this was a quick video, which is good because it's time for me to go take care of my kids. So I hope you hit a home run like my daughter Ellie here. And enzymes, I feel like, will be the most clear part to you. And we'll do a Glencoe virtual lab with them. Take care, students, and study hard.